Okay, so the structure of the RNA and how can RNA fold upon itself. So if you go on seeing the structure of RNA and DNA, you can say that both of them are quite similar to each other. However, there are certain differences that makes RNA more simpler as compared to DNA. DNA essentially is double-stranded in nature, however, RNA is just single-stranded. And also, other than that, RNA is basically a basic form of DNA that is usually used for the transfer of information from one place to another. And hence, it is the genomic material of more primitive organisms like viruses. However, after further research and looking into the matter, we found out that RNAs are not as simpler as we thought. In order to compensate for their lack of complex structure, they can fold around themselves and have many different structural configuration, like a knot-like formation, and sometimes they can also act like enzymes. Now, despite of being single-stranded, RNA molecules often fold around themselves to adopt stem loop-like structures. Now, these stem loop-like structures can have internal loops and internal bulges. Sometimes there also might be knots. So we can consider a RNA strand as a string that can fold all around itself, that can have bulges and can have knots. But why does this happen? Now, it's important to note that these bulges and these loops are actually helpful for the RNA because sometimes, let's take an example of the loop called as UUCG. So basically, a normal stem loop, we have a tetraloop or a four-base loop called as with the sequence UUCG. Now, this helps for the stability of the RNA. Well, this was for the stability, but sometimes there's also the formation of knot-like structures called as pseudoknots, which are basically between sequences that are not matching with each other. Although RNA has this ability of forming primary, secondary, and tertiary structures, sometimes it can become a bit more complex because adenine in the RNA molecule can form two hydrogen bonds simultaneously with two uracils, which might lead to this entire complex to be more tangled. Also, in RNA molecule, the guanine and the uracil can be paired by the formation of two hydrogen bonds. Now, these two hydrogen bonds are formed between the sixth oxygen atom of the guanine molecule and the third nitrogen atom of the uracil molecule. And in the case of the other hydrogen bond, it's between the first nitrogen atom of the guanine molecule and the second oxygen atom of the uracil molecule. So, because of the non-standard pairing between the bases, it's possible to observe that the base pairings between the two RNA strands are quite versatile. And this is the reason why RNA is not the repository of biological information. However, with the exception of mRNA and other non-coding proteins. But in order to compensate, they have quite complex structures. The process of translation is quite important for a cell because in a rapidly growing cell, about 80% of the cell energy and 80% and 50% of the cell's dry weight is used for the process of translation. And the process of translation is basically taking a protein and formation of a protein from an amino acid polypeptide chain. The machinery that is responsible for translating the language of an mRNA, which is the template, into a normal protein includes the amino acid tRNA synthase, the mRNA, the tRNA, and the ribosome. Together, they accomplish the task of transforming a 4-base code of written language into a 20 amino acid sequence. So, what is an mRNA? An mRNA is a template or reference material that is used in order to translate, in order to put in amino acids. Now, this template material or the mRNA has something called as the open reading frame. The open reading frame is the path through which translation takes place. By the ribosomes, Translation always begins in the direction of 5' to 3', and in an open reading frame, there's always a start and a stop codon. For a typical eukaryotic cell, the start codon is always AUG. That is placed at the initial point of the open reading frame. And at the end of the open reading frame, we have 3 types of stop codons for a typical eukaryotic cell. They are UGA, UAG, and UAA. And mRNA can have multiple open reading frames or a single open reading frames. Usually in the case of eukaryotes, we have one open reading frame. And in the case of prokaryotes, we have multiple open reading frames. This is because an RNA molecule can be monocistronic as in the eukaryotes, and polycistronic in the case of prokaryotes. Polycistronic types of mRNA have this inherent ability to translate many different proteins from one single mRNA molecule.